that's exactly what we saw in the youngest kids who were enrolled in this study. This was in the 5 countries in Asia where they enrolled kids very young. You can see kids who were 2 to 5 years of age at the time of vaccination. In year 3 of the study, which is 2 years after they've completed their vaccination series, they had a 7.45 relative risk of hospitalization due to dengue if they were vaccinated 3 years previously. So the vaccine gave them 7.45 relative risk of hospitalization for dengue if they were subsequently infected with dengue. You can see that as you got older, that risk went down. So the big question was, is this an age phenomenon? Is this a serostatus phenomenon? Now they didn't draw pre-vaccination blood samples in everyone, and only about 10% of volunteers got a pre-vaccination blood sample. So they couldn't do this analysis on pre-vaccination blood samples, but they were able to develop an assay later on to try to answer this risk, and this is one of the requirements that SAGE set. So what we saw was variable efficacy based on serostatus, based on age, based on serotype. Very good efficacy against hospitalized dengue and dengue and severe disease, but there was an increased risk of severe disease in the youngest of kids. And what we found out later through an assay that was developed was that that risk was determined to be serostatus at vaccination. So now we know on an individual level, if you've never had dengue, you're at elevated risk for more severe disease, but on a population level, we can have a very good impact against hospitalization and severe disease. So this vaccine was in fact licensed, and in April of 2016, it was launched in the Philippines. It was launched following guidance by SAGE that said it should only be used in highly endemic areas in kids 9 or older. So in April of 2016, it was launched. In November of 2017, Sanofi relabeled the vaccine to be used only for those children who already had dengue. And then in February, it became a national scandal. And it was really devastating across the Philippines, not just for dengue vaccination, but what you can see here is that all vaccinations, the confidence in vaccines fell dramatically in the Philippines because of this episode. And it wasn't that really there was a lot of new information out there at the time. It was how this information was perceived and how the relabeling of the vaccine for only those who already had dengue was perceived by the public. But it was devastating. The Philippines is still recovering, trying to recover vaccination rates because of this episode. So what did we learn? We learned that neutralizing antibody is not predictive of vaccine success, but that we need to test for serostatus before vaccination. And now the recommendations from regulatory authorities is that everybody needs to have a pre-vaccination blood draw. We also know that, or we think that CD8 T cells may be important in the mitigation of disease of dengue, and that we now think that it's important to have CD8 epitopes in the vaccine. And we also learned, very importantly, as we've all learned from COVID and many other vaccine incidents, is that messaging and transparency are very, very important. We have to gain and we have to maintain the public trust if we're going to use these vaccines. So I'm going to move on now to TAK-003. This was a phase 3 clinical trial that was conducted in numerous countries in Asia and Latin America. They enrolled more than 20,000 people. 27.6% were dengue naive at the time of enrollment, and that's important when we look at the different results of these trials, because these vaccines behave differently for the most part in people who've already had dengue and those who've not already had dengue. So this vaccine has excellent efficacy in year 1 against both virologically confirmed dengue of any severity as well as hospitalized dengue, and it performed well in year 1 in both those who were seropositive and seronegative. What we see as we go down, this is year 1, this is year 2, year 3, and then cumulative data for the first 3 years of the study. And what we see here in seronaives is that efficacy against virologically confirmed dengue declines over time, and in seronegatives, and efficacy against hospitalized dengue declined over time in seronaives, and was not significant in year 3 of the study. Now this was driven by a particular serotype, dengue serotype 3, and that's something that we're going to talk a little bit about. But I think it's important to remember that these numbers, these serotypes. You can have efficacy against different serotypes, and that can affect your overall efficacy, and it's important to note. So what you see here is even though in year 1, you had 82% efficacy against virologically confirmed dengue in seropositives and 74.9% in seronaives, what you see is that is varied by serotype. So this high efficacy is really driven by dengue 2 over each of the 3 years. You can see that very good efficacy against dengue 2 is maintained over the 3 years. And again, this is the only full-length dengue vaccine in the vaccine. The other components are chimerics. We do not see significant efficacy against dengue 3 or dengue 4 in people who have not had dengue before, and we're losing efficacy to dengue 1 over time in seronaives. So we're seeing a difference in the protective efficacy of the vaccine in seronaives to 3 of the 4 dengue serotypes. When we look at year 3, and again, this was driven by dengue 3. This was driven by dengue 3, and particularly hospitalized cases of dengue 3 in one country. But what you can see is no overall efficacy against hospitalized dengue 3 in seropositives or seronegatives in year 3 of the study. And in seronegatives, you can see that the confidence intervals are quite large. Now, part of that is there are a limited number of cases of dengue 3, but it is a signal that people are currently watching, and SAGE has asked for additional information to try to get a better handle on what this may be due to. So we have, again, excellent efficacy against hospitalized virologically confirmed dengue in dengue-naive subjects for 2 years post-vaccination. But again, we're seeing varying efficacy by serotype and serostatus, and declining efficacy over time, particularly in those who are seronaive. There was a signal, although it was not statistically significant, for dengue 3. There were excess cases of dengue 3 in year 3 of the study, hospitalized dengue 3, again, driven really by hospitalization in one country. But again, we're seeing an overall population benefit. You can have a huge impact with the protection against hospitalization and severe disease, with the potential individual risk, and how do we balance that? This was SAGE met less than a year ago, September of 2023, to discuss this. And what they decided was that they're going to recommend introduction to children aged 6 to 16, even though the study was down to age 4, in settings with high seroprevalence of dengue, which was defined as a seroprevalence of 60% or greater by age 9. This is a 2-dose schedule, 3 months apart. It did not recommend, SAGE did not recommend vaccination of children 4 to 5 years of age because of the low performance of the vaccine in this age group in both seropositive and seronaives. They recommended post-licensure studies to provide more precise estimates of the effectiveness and risk profile against dengue 3 and dengue 4 in seronegative persons. And they said travelers with previous dengue vaccine should be vaccinated. Travelers without previous dengue vaccine should be told that the vaccine may not work against dengue 3 and 4. There is no testing requirement prior to vaccination for this vaccine. The company with its BLA with the FDA, so this vaccine, as Gabrielle mentioned, will not be licensed in the US in the near future. We'll see if the company decides to resubmit their BLA to the FDA, but we won't have this vaccine available in the US for several years to